Welcome, curious minds, to the deep dive. Imagine this complex molecular economy running inside you right now, recycling precious resources, generating fuel, even building critical brain chemicals. Today, we're unlocking the secrets of amino acids, the very currency of this economy, and revealing how their intricate metabolic pathways dictate pretty much everything from your energy levels to your overall health. You might primarily think of amino acids as, well, just building blocks for proteins.
Yeah, that's the common view.
But their metabolic pathways are actually far more diverse, a surprisingly complex biochemical ballet, you could say, crucial for everything from energy production to proper brain function.
Exactly. And what's truly remarkable, and maybe often overlooked, is how even a seemingly small defect, like one tiny glitch in one of these intricate pathways, can ripple through the entire system, leading to really significant clinical consequences. Our mission today is to kind of demystify these pathways for you, highlight the most important nuggets of knowledge, and connect the dots to real-world health implications, things you might actually encounter or study.
Right, so whether you're grappling with biochemistry for an exam, prepping for a medical meeting, or just have that insatiable curiosity about how your body performs its daily miracles, get ready for an engaging tour. We're going through the synthesis and degradation of amino acids, and we'll be focusing on a key chapter from Marks Basic Medical Biochemistry, a clinical approach to kind of guide us through this fascinating journey. Okay, to begin, let's lay some foundational knowledge. We often hear about the major enzymes, right? But what about their crucial sidekicks, the coenzymes? These are the essential helpers in amino acid metabolism, aren't they?
They absolutely are. Indispensable. Think of them as the unsung heroes, really. Three in particular play starring roles.
Hmm.
First, there's pyridoxal phosphate, or PLP, comes from vitamin B6.
Okay, B6.
Yeah. This molecule is like the metabolic maestro, orchestrating nearly every major amino acid transformation. We're talking reactions like transamination.
Which is like swapping amino groups around.
Exactly like a molecular swap meet for amino groups.
Yeah.
But also deamination, decarboxylation, elimination reactions. PLP is, right at the heart of almost every amino acid pathway. Next up, we have tetrahydrofolate, FH4. That's from the vitamin folate.
Folate.
FH4 is the body's main carrier for one-carbon groups. This is pretty critical for both breaking down certain amino acids like serine and histidine, and for building new ones like glycine. And finally, tetrahydrobiopterin, BH4.
pH4.
This cofactor is vital for specific ring hydroxylation reactions. That's where molecular oxygen gets precisely incorporated. Think crucial conversions like changing phenylalanine into tyrosine.
Okay.
And it's also a key player in synthesizing important neurotransmitters in your brain.
Wow. So these aren't just minor characters in the metabolic drama at all.
Not at all.
They're truly central to the whole narrative. Now, let's talk about how amino acids are actually made within our bodies.
Well, of the 20 common amino acids, humans can synthesize 11 of them. We call these non-essential because, well, our body doesn't essentially need them from our diet.
Right? We can make them.
We can make them. The other 9 are essential. You got to get those from food. What's particularly fascinating, though, is that the amino acids we synthesize aren't just destined for protein building. They're often diverted to create other vital nitrogen-containing compounds.
Like what?
Well, for instance, glycine contributes to porphyrins for hemoglobin. Glutamate is a precursor for important neurotransmitters. Aspartate helps build purines and pyrimidines.
The DNA and RNA building blocks.
Exactly, the building blocks of DNA and RNA.
And it's really clever how our bodies source the basic carbon skeletons for these non-essential ones. Many of them get their initial structure right from intermediates of glycolysis and the TCA cycle, meaning they ultimately originate from glucose, right? A very efficient reuse of metabolic material.
It really is.
Yep.
But there's a unique twist for tyrosine and cysteine. They actually require an essential amino acid for their synthesis.
Oh, interesting.
Yeah, tyrosine needs phenylalanine, and cysteine gets its sulfur from methionine, even though its carbons still come from a glucose-derived intermediate like serine.
Huh. And the body must regulate this pretty tightly.
Oh, incredibly tightly. Think of it like a metabolic thermostat. As the concentration of a free amino acid rises, it can actually inhibit a key enzyme early in its own production pathway.
Feedback regulation.
Exactly, feedback regulation. It ensures a perfect balance, prevents wasteful overproduction.
That's a powerful internal control. Okay, we've talked about making them. What happens when the body needs to break them down? Are the degradation pathways just the reverse of synthesis? Or are they entirely different systems?
Generally, they're distinct. Degradation pathways usually follow different routes than biosynthesis.
Ah, okay.
And this allows for separate regulation of anabolic, building up process.
And catabolic breaking down.
Right. Catabolic processes.
Hmm.
Gives the body much finer control. When amino acids are degraded, their carbon skeletons can have several fates, depending on your body's physiological state,
Mm-hmm.
even the specific tissue involved.
Such as? What kind of fates?
Well, they can be fully oxidized to carbon dioxide from energy, or they can be converted into precursors that the liver can then readily turn into glucose.
Okay.
These glucose precursors are often intermediates of the TCA cycle, like α-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, or even pyruvate.
Mm-hmm.
Amino acids, whose carbons can form glucose, are classified as glucogenic.
Glucogenic. Makes sense glucose generating.
Precisely. Others are converted directly to acetyl-CoA or acetoacetate. Those molecules can form ketone bodies or fatty acids, so these are known as ketogenic amino acids.
Ketogenic.
And just to add another layer of complexity, some amino acids can actually be both glucogenic and ketogenic.
Wow. Okay, so if you're fasting, say, the liver acts like this incredible factory, taking those amino acid carbons and turning them into vital glucose or ketone bodies for fuel. But in a fed state, the same carbons might get channeled into making glycogen or fat for storage.
Right. It adapts. That's why the liver is so central. It's the grand central station of amino acid metabolism, really. The only tissue that has all the pathways for both synthesis and degradation.
Okay, let's dive into some specific amino acids now. Maybe start with those that get their start from glycolysis intermediates.
Sounds good. This group includes serine, glycine, cysteine, and alanine. When these guys are degraded, their carbons usually feed back into pyruvate or other intermediates in the glycolytic pathway, meaning they're primarily glucogenic. They can become glucose.
Makes sense. Let's start with serine. How does this one get made and broken down?
Okay, serine is cleverly built from 3-phosphoglycerate. This is a direct glycolysis intermediate.
Right.
It goes through a sequence of chemical transformations. It involves oxidation, an amino group transfer, and then taking off a phosphate. It's like converting a basic sugar building block into a more specialized amino acid.
And breaking it down.
For degradation, serine typically undergoes transamination, eventually leading back to pyruvate. Or there's a direct elimination of water by an enzyme called serine dehydratase, also forming pyruvate.
Okay. And glycine. You mentioned this one is surprisingly involved in some serious clinical issues.
Indeed it is. Glycine is mainly synthesized from serine. It's a reversible reaction, and fittingly, it needs both FH4 and PLP.
Those cofactors again.
Yep. In its degradation, the conversion of glycine to a molecule called glyoxylate is critically important clinically, because glyoxylate can then be oxidized to oxalate.
Oxalate. Kidney stones, right?
Exactly. Here's where it gets really impactful. Oxalate is sparingly soluble. It can precipitate in kidney tubules, leading to the formation of painful kidney stones, often calcium oxalate stones. And there's a genetic defect, a defect in the enzyme that normally converts glyoxylate back to glycine, which causes primary oxaluria type I, or PH1.
Oh, wow.
Yeah, this leads to excessive oxalate accumulation throughout the body and, sadly, often results in renal failure.
That's serious. What about cysteine?
Cysteine's carbons and nitrogen come from serine, but its sulfur is unique. It's donated by the essential amino acid methionine.
Ah, okay. Needs methionine.
Needs methionine sulfur. Serine reacts with homocysteine, which comes from methionine, to form an intermediate called cystathionine.
Right.
This reaction is catalyzed by cystathionine β-synthase. Then, cystathionine is cleaved to cysteine and α-keto butyrate by another enzyme, cystathionase. Both of these crucial enzymes, by the way, require PLP.
PLP again. So if your dietary intake of methionine is low, cysteine, which usually isn't essential.
It can become essential, exactly. That's a fascinating metabolic nuance, isn't it?
It really is.
Cysteine also regulates its own production through that feedback inhibition mechanism we talked about earlier. Clinically, you might see something called cystathionuria, where cystathionine shows up in the urine. This can happen in premature infants, or maybe due to a deficiency in cystathionase, or even a vitamin B6 deficiency. It's often considered relatively benign, usually doesn't cause severe health problems. However, not all issues with cysteine are benign. Problems with cysteine transport lead to much more serious disorders.
Like cystinuria.
Exactly, like cystinuria. There, a defective kidney transport protein means cysteine, and also lysine, arginine, and ornithine aren't properly reabsorbed. Cysteine then accumulates and, being poorly soluble, forms kidney stones.
Okay.
And then there's cystinosis, which is different again. It's a defective lysosomal carrier. This causes cysteine to accumulate inside lysosomes, forming crystals that damage cells. It often leads to renal failure in children, maybe by age 6 to 12. Very serious.
This ties directly into a clinical case for our listener, Horace S., a 14-year-old boy. He had some unusual symptoms pointing right at these pathways.
Yes, exactly. Horace presented with sudden weakness, dislocated lenses in his eyes, and a slight intellectual disability. His lab work showed very high methionine and homocysteine levels,
High methionine and homocysteine.
but notably low cysteine levels.
Low cysteine.
Yeah, this pattern strongly points to a problem in methionine degradation, specifically homocystinuria. This is often caused by a deficiency in that enzyme we mentioned, cystathionine VOCITs.
The one that makes cystathionine from homocysteine and serine.
Precisely. Or sometimes it can be issues with FH4 or vitamin B12 metabolism, which are needed to recycle homocysteine back to methionine. Elevated homocysteine is a serious concern. It's linked to cardiovascular disease, blood clots, damage to vascular cells.
Not good. And finally, for this group, alanine, also from glycolysis.
Right, alanine is produced directly from pyruvate by alanine amidotransaminase, or ALT. It's a readily reversible reaction.
ALT, that's a common liver enzyme test.
It is indeed. Alanine is considered the major glucogenic amino acid because it acts as a critical transporter. It ferries nitrogen from various tissues, especially muscle, to the liver. There, the liver can use the carbon skeleton for gluconeogenesis, making glucose, and the nitrogen gets safely excreted as urea.
Okay, let's shift gears. Next, we move to amino acids that connect with the TCA cycle. Sounds like another prime example of the body recycling its resources.
Absolutely. More connections. There are two main groups here. Those derived from α-ketoglutarate and those from oxaloacetate.
Let's start with the α-ketoglutarate group. Glutamate, glutamine, proline, arginine, and histidine.
Okay. Glutamate itself comes directly from α-ketoglutarate, so all its carbons can ultimately be derived from glucose. It's a very versatile amino acid. It acts as a precursor for many others, like
Glutamine, proline.
glutamine, proline, ornithine, arginine, and it also forms a crucial part of glutathione.
The antioxidant.
The incredibly important antioxidant, yeah, protects your cells from damage. Then glutamine. It's made from glutamate by an enzyme called glutamine synthetases. This enzyme has a really unique and vital role. It's one of only 3 human enzymes that can fix free ammonia into an organic molecule.
So a detoxifier.
A crucial detoxifier, yes. It's particularly important in the kidney. There, it gets converted back to glutamate, releasing ammonia, which helps buffer acids for excretion in the urine, helping regulate your body's pH balance.
Clever system. And proline, that one has a distinctive ring structure.
It does. Proline synthesis starts with glutamate, which undergoes a series of transformations, including cyclization and reduction, to form that ring. Its degradation is essentially the reverse process. Proline, with that unique cyclic structure, is particularly important for collagen.
Connective tissue.
The most abundant protein in our body, yeah. Provides crucial structural integrity to tissues.
What about arginine?
Arginine is synthesized from glutamate via ornithine, which is an intermediate of the urea cycle.
Right, the urea cycle.
And here's a surprising metabolic twist. Arginine, though usually synthesized, can actually become essential during periods of rapid growth or maybe recovery from trauma.
Why is that?
Because the amounts produced by the urea cycle might simply be insufficient to meet the body's increased demands during those times. It really highlights the dynamic nature of essential nutrients, doesn't it?
It really does. Conditional essentiality.
Exactly. It's degraded by the enzyme arginase, yielding urea and ornithine, completing that cycle link. And lastly for this group, histidine. Although it's an essential amino acid, humans can't synthesize it. Its degradation pathway eventually yields glutamate. But before that, histidine is also the precursor for vital compounds like histamine.
Allergies, stomach acid.
Right, a key player in immune responses and stomach acid secretion. Shows its diverse roles beyond just protein.
Okay, moving to the oxaloacetate group, aspartate and asparagine.
Simpler group here. Aspartate is formed by a transamination reaction involving oxaloacetate, another TCA cycle intermediate. It's readily reversible. Asparagine is then formed from aspartate. Glutamine donates the nitrogen for its amide remainder group, and the enzyme asparaginase converts it back to aspartate.
Asparaginase. That reminds me of a really clever clinical application in cancer treatment.
Indeed. Very clever. Certain types of tumor cells, especially some leukemic cells, have a high requirement for asparagine to fuel their rapid growth.
They can't make enough themselves.
Often they can't, or not enough. So asparaginase is actually used as an anti-tumor agent. It works by breaking down asparagine in the blood, converting it to aspartate. This effectively depletes the amount of asparagine available externally, starving those rapidly dividing tumor cells that depend on it.
That's brilliant.
It's been a mainstay treatment for decades for acute lymphoblastic leukemia.
Okay, let's broaden our view now to other breakdown products, amino acids connecting to different parts of the energy landscape.
Absolutely. The carbons of aspartate, for example, can also form fumarate within the urea cycle, showing more interconnectedness. And as we'll touch on more, phenylalanine and tyrosine, the aromatic amino acids, also eventually yield fumarate, along with acetoacetate.
And those that form succinyl coenzyme A, which ones are they?
That would be methionine, valine, isoleucine, and threonine. These are initially degraded to an intermediate called propional-CoA,
Propional-CoA.
which then gets converted to succinyl-CoA. This conversion is a critical process, and it requires both biotin and vitamin B12.
B12 again.
Yep. And interestingly, it's the exact same pathway used to metabolize odd-chain fatty acids, another link between metabolic pathways. And to revisit methionine quickly, it's first converted to acetylcysteine, or SAM,
The methyl donor.
the major methyl group donor in the body, yes.
Yeah.
After donating its methyl group, it becomes acetylcysteine, SAH, and then homocysteine.
Yeah.
Now, methionine can be regenerated from homocysteine that requires both FH4 and vitamin B12. Or, homocysteine can provide its sulfur for cysteine synthesis, using those PLP-dependent enzymes we mentioned earlier. The remaining carbons of homocysteine are then metabolized, eventually leading to succinyl-CoA.
Right. And getting back to Horace S briefly, his doctor found very high methionine and homocysteine, but normal B12 and folate levels.
Crucial finding.
Yeah, because it helped confirm his homocystinuria was due to a deficiency in cystocyanine β-synthase.
The enzyme linking homocysteine to cysteine synthesis.
Exactly. Rather than issues with methyl group transfer involving B12 or folate. This specific defect leads to those serious issues: blood clots, skeletal problems, intellectual disability.
And treatment often involves a low methionine diet, and importantly, for about half of these patients, high oral doses of vitamin B6.
B6, the precursor for PLP.
Exactly. It helps boost the activity of any residual cystathionine epistatase enzyme they might have.
Makes sense. What about threonine, also formed succinyl-CoA?
Yes. Threonate is primarily degraded in humans by a PLP requiring dehydratase enzyme.
Yeah.
This generates ammonia and α-ketoglutarate, which then funnels into that same propional-CoA pathway, ultimately forming succinyl-CoA, just like methionine.
Okay. And the branched-chain amino acids, valine and isoleucine, they sound like prime candidates for muscle fuel.
They certainly are. Valine, isoleucine, and leucine, the three branched-chain amino acids, are all essential. And they're crucial fuels, especially in muscle tissue during fasting or exercise. Their degradation not only generates energy, but also replenishes TCA cycle intermediates, which is important for sustained energy production.
How are they broken down?
They initially undergo transamination, removing the amino group. Then they go through what we call oxidative decarboxylation.
Okay.
It's essentially a two-in-one reaction where a carbon is lost as CO2 while the molecule is oxidized, often generating energy carriers like NADH. This step is catalyzed by the ketoacid dehydrogenase complex.
A complex, like pyruvate dehydrogenase.
Very similar, yes. A multi-enzyme system. And both valine and isoleucine ultimately produce succinyl-CoA from parts of their carbon skeletons. Isoleucine also produces acetyl-CoA, making it both glucogenic and ketogenic. Valine is purely glucogenic.
Given how critical this aceto acid dehydrogenase complex is, for all three branched-chain amino acids, I imagine its malfunction could lead to some very serious conditions.
Absolutely. That's precisely what causes maple syrup urine disease, or MSUD.
MSUD.
It's caused by defective branched-chain aceto acid dehydrogenase. This leads to the accumulation of these branched-chain amino acids, leucine, isoleucine, valine, and their corresponding aceto analogs in the blood and tissues.
And the consequences.
Severe neurological complications, developmental delay, and distinctively a maple syrup or burnt sugar odor in the urine, which gives the disease its name. It's a very difficult condition to manage because you have to restrict 3 essential amino acids in the diet. Also worth noting, a severe thiamine deficiency can affect this complex, since thiamine pyrophosphate is a required cofactor. This can lead to accumulation of the α-keto acids, sometimes seen in patients like Al-M, who suffer from chronic alcoholism leading to poor nutrition.
Right, thiamine deficiency affecting energy metabolism broadly. Okay, finally, let's talk about amino acids that primarily form acetyl-coenzyme A and acetoacetate, the ketogenic ones.
Okay, 7 amino acids are classified as ketogenic in some way. Interestingly, isoleucine, threonine, phenylalanine, tyrosine, and tryptophan are actually both glucogenic and ketogenic.
Ah, the mixed ones.
Right. Their carbons can contribute to both glucose and ketone bodies or fatty acids. However, leucine and lysine are strictly ketogenic. They only produce acetyl-CoA and/or acetoacetate, and therefore cannot contribute at all to net glucose synthesis.
Strictly ketogenic. Got it. Let's focus on phenylalanine and tyrosine for a moment, as this directly relates to our other clinical case, Patria-Y.
Right. Phenylalanine, an essential amino acid, is converted to tyrosine by the enzyme phenylalanine hydroxylase, or PAH. This reaction needs molecular oxygen and that cofactor BH4 we mentioned earlier.
BH4 again?
Yep. Tyrosine, which is now non-essential if you have enough phenylalanine, is then further oxidized through a series of steps to yield both acetoacetate, ketogenic, and fumarate, glucogenic. So tyrosine is one of those mixed ones.
And a defect in this first step, in PAH, causes phenylketonuria, or PKU.
Yes, exactly. Patria-Y, the infant in the case study at 4 months old, showed developmental delays, tremors, seizures, and that distinct musty or mousy odor in her diaper caused by phenylacetate accumulation.
Oh, dear.
A screening test revealed excess phenylalanine. Subsequent testing showed her plasma phenylalanine was severely elevated, over 20 milligdl. Normal was around 1 milligdl. And PAH activity in her liver was less than 1% of normal. This confirmed classic PKU, an autosomal recessive disorder.
What are the devastating consequences if PKU is left untreated? It sounds critical to catch it early.
It is absolutely critical. If PKU isn't recognized and treated, usually within the first month of life, the high phenylalanine levels cause severe problems. They interfere with crucial amino acid transport into the brain and also neurotransmitter synthesis. This leads to irreversible intellectual disability, delayed psychomotor maturation, seizures, a really tragic outcome if missed.
So those newborn screenings like the Guthrie test or newer methods are clearly paramount.
Absolutely vital. Early detection allows for immediate treatment. That's typically a strict low phenylalanine diet using special medical formulas that provide other amino acids, but very little phenylalanine. Lifelong dietary adherence is critical, yes. Even for adults, high phenylalanine can cause neurological issues. And it's especially crucial for pregnant women with PKU to maintain strict control to avoid severe damage to the developing fetus, known as maternal PKU syndrome. Supplementation with large neutral amino acids can sometimes help too, by competing with phenylalanine for transport into the brain. And there are other related disorders too, like alkaptonuria, where a later enzyme in tyrosine degradation, homocystinuria oxidase, is defective. This leads to dark urine upon standing, and later in life, severe arthritis due to pigment depositions in joints.
Dark urine, interesting.
Yeah. Also, various tyrosinemias exist due to specific enzyme defects further down the tyrosine metabolism pathway. Some severe forms can cause liver failure and require liver transplantation. It's also worth quickly mentioning malignant hyperphenylalemia.
Malignant.
Yeah, sounds bad. Here, the PAH enzyme activity itself is normal, but the issue lies in regenerating or synthesizing the cofactor BH4.
Ah, the cofactor again.
Exactly. These patients need BH4 administration and often neurotransmitter replacement therapy, because BH4 is also essential for synthesizing dopamine and serotonin in the brain. The dietary restriction alone isn't enough.
Complex interactions. What about tryptophan, another mixed one?
Yes, tryptophan is both glucogenic and ketogenic. During its degradation, it yields alanine, glucogenic, formate, which enters one-carbon metabolism, and acetyl-CoA, ketogenic. Its distinctive indole ring structure can also be used through a separate pathway to produce NAD+ and NADP+ suc.
The niacin coenzymes.
Exactly. So tryptophan intake actually spares our dietary need for niacin, the B vitamin.
So a deficiency in both niacin and tryptophan could have significant consequences.
Indeed, if both levels are insufficient, it can lead to pellagra.
Pellagra, the 3Ds.
Often characterized by the 3Ds.
Where?
Dermatitis, diarrhea, dementia, and potentially a fourth B, death if untreated. Tryptophan degradation also requires vitamin B6, PLP. A B6 deficiency can lead to the excretion of an intermediate called xanthorenic acid in the urine. And finally, leucine and lysine, strictly ketogenic.
Right.
Leucine is a third branched chain amino acid. It forms acetyl-CoA and acetoacetate, with no contribution to glucose synthesis. Lysine is degraded through a pretty complex pathway, also ending strictly in acetyl-CoA.
Wow, what an incredible journey, seriously. From the essential coenzymes just orchestrating these transformations to the complex dance of metabolic pathways and the profound, often devastating impact of tiny errors in that biochemical machinery.
We've truly seen how amino acids are so much more than just protein building blocks, haven't we?
Absolutely.
They're incredibly versatile molecules. They contribute to our energy reserves. They create key compounds like the antioxidant glutathione. They even profoundly influence brain function and overall health. And understanding that distinction between glucogenic and ketogenic pathways and the specific roles of those key cofactors like PLP, FH4, and BH4, it really is foundational to understanding human biochemistry.
Yeah, it really brings it together. And the clinical examples like Petria Y's PKU and Horace S's homocystinuria, they really drive home the importance of these pathways, don't they? And also the ingenuity of things like early detection and dietary intervention. It's a testament to the delicate balance within our biochemistry and how understanding these pathways lets us identify and treat these potentially devastating diseases.
Absolutely. The body's ability to synthesize, degrade, and regulate all these pathways is incredibly sophisticated. And disruptions, as we saw, can manifest in such diverse and challenging ways. It truly underscores that, every biochemical reaction plays a crucial role in overall health. A single enzyme defect can just have these cascading effects.
So what does this all mean for you listening? Well, next time you think about amino acids, maybe remember this deep dive into their incredible dynamic metabolic lives. Consider how these seemingly small molecules have such far-reaching effects across your entire body, connecting your diet, your energy, and your health in ways you might not have even imagined. Thank you so much for joining us on this deep dive into amino acid metabolism. We genuinely hope you've gained valuable insights.
And we hope this discussion maybe sparks some further curiosity for you to explore this fascinating and really vital subject even more.
This has been The Deep Dive. From the Deep Dive team, thank you for listening.